Dear aspirant, in this course, we are going to see one of the important industry in pharmaceutical sector, bioequivalence and bioavailability centers. This is important because the bioavailability and bioequivalence studies are a critical steps to succeed the generic formulation development. So we will see how the bioequivalence industry works particularly in India. So in bioequivalence industry, these studies are carried out in bioequivalence centers, and these centers are either part of pharmaceutical companies or they can be independent contract research organizations. Means the pharmaceutical companies can have a separate department to conduct the pharmaceutical development programs like bioequivalence and bioavailability centers, or these bioequivalence studies can be carried out by an independent contract research organization. And these bioequivalence and bioavailability studies are mostly part of the generic formulation development, which I already explained. And sometimes this can be part of the new drug development or new dosage form development, where we assess the bioavailability and pharmacokinetics of the different formulations. And in most bioequivalence studies, the generic formulation under development, that is a test product, is compared with the already marketed reference product, which can be marketed either in Western countries or sometimes in the local country. So this particular slide in a bioequivalence study schematically represents a comparison of test product with a reference product. So the test product here is in a pink color and the reference product in white color tablets, and this can be either in the same shape or form or in a different form, different shape. But ideally, in case of bioequivalence, the qualitative and quantitative composition of the drug product in most of the times try to keep as same as possible. That is called pharmaceutical equivalence. If both qualitative and quantitative composition of the drug product is same, it's called pharmaceutical equivalence. And the bioequivalence studies, we are comparing the safety and efficacy in terms of the bioavailability. The next diagram in terms of the pharmacokinetic representation is explaining the safety and efficacy comparison for the test and reference product. In this particular diagram, we can see the test product plasma drug concentration profile as well as the reference product plasma concentration drug profile, both in comparison. And we see that whether both plasma profiles are very close to each other and statistically can be compared to show the bioequivalence or not. So this is how the bioequivalence will be concluded in terms of pharmacokinetics and statistical comparison. In this slide, we are seeing the list of bioequivalence centers in India published by the Indian Regulatory Agency CDSEO. There are 79 facilities already reported in the published list of CDSEO. Some of the sample names are Asian Drug Research Solutions in Hyderabad, Synapse Labs Private Limited in Pune, Par Biosciences Private Limited in Chennai, Glenmark Research Center in Mumbai, Cliantha Research Limited in Ahmedabad. In these, Synapse, Cliantha, Eisent they are independent contract research organization. They are not associated with pharmaceutical company. Otherwise, this Par Biosciences Private Limited is part of Endo pharmaceutical company, and also this Glenmark Research Center is part of Glenmark Pharmaceuticals Limited, which is again a pharmaceutical company. If you divide a bioequivalence facility into different departments in terms of activities, you can see that there will be a clinical department, clinical facility, there will be a bioanalytical department, then there will be data analysis department where you do the pharmacokinetic and statistical analysis, then there will be a quality assurance department, there will be a department which do the scientific writing, which includes study protocol writing and the study report writing, then there will be a HR and administration department, then there will be a logistics and stores department which carry out the entire supply chain management for the studies. In a bioequivalence facility, the major part of the clinical facility will be divided into different areas. So the first area is the screening facility in the clinical facility. The screening facility generally screen the volunteers or subjects to be recruited for the bioequivalence studies. So the screening facility, the volunteer comes and they will be screened for their health and fitness for the study. As part of the health and fitness, there will be a pathological lab which evaluate the hematology, biochemistry, serology, urine analysis, electrocardiogram, etc. And coming to the housing and study facility, where it comprises the beds, intensive care unit, recreational area for the subjects and volunteers, washrooms, and dining area. Then finally, the collected samples, the pharmacokinetic samples, will be processed and stored in the sample processing and storage area, which comprises deep freezers as well as refrigerator, centrifuge, etc. In bioanalytical facility, we will have the major instruments like liquid chromatography mass spectrophotometers, LCMS or HPLC. Then we will have centrifuge, deep freezers, extraction instruments like SP, cartridges, etc. Then there will be microbalance to weigh the standards and samples. Then there will be a sample processing area where we do the sample processing for separating the drug from the sample matrices, and there will be vortex machines and other support equipments. Coming to the data analysis where we do the pharmacokinetic and statistical analysis, we generally do by the softwares present in the computers. The software generally used are like Winolin or statistical analysis software like SAS. So basically we are trying to reiterate that bioequivalent studies are clinical studies. So they are basically part of the initial pharmaceutical development in case of new drug development, where we generally understand the pharmacokinetics of a drug product. So this comes under phase one, early phase one studies or during the early phase two development during the drug discovery. So these bioequivalent studies can be called as clinical studies, part of the clinical development. And in case of generic formulation development, these clinical studies are carried out in healthy human subjects to assess the pharmacokinetics. In case of drug discovery, it can be either in healthy human or it can be in patients also. So sometimes, in case of generic development also, we use the patients as volunteers because the drugs are cytotoxic or unsafe in nature for the healthy subjects. So, for example, this cancer, all the cancer drugs are being carried out in patients because of the cytotoxic nature. Then these healthy subjects has to prove their healthiness through the screening tests. So that's where we explain the pathological test what we carried out in the bioequivalent studies. And the bioequivalent facilities are more in abundant in India because the volunteer population is extensive and very popular in culture. In the coming couple of slides, we are going to see the expertise and professionals involved in the bioequivalent studies. So coming to the clinical, there are several professionals involved in the bioequivalent studies, including medical doctors who generally principal and clinical investigators lead the clinical facility qualified with the MBBS or MD degree. And these medical doctors will be assisted by nurses who take care of the subject safety and blood sampling. Their qualification will be generally diploma or degree in nursing. And also to collect the blood samples, the bioequivalent studies employs phlebotomists who have the qualification of DMLT or by training they acquire these phlebotomy skills. Then there will be clinical research associates. So the clinical research associates can be having any kind of degree, including pharma, nursing, etc. And also there are several CRAs with who have the life science graduates, basically from the biochemistry and biotechnology background. And apart from this, we have ECG technicians who takes the ECGs for the subjects. Sometimes these are carried out by nurses. And there will be volunteer recruitment officers. These are from any kind of graduates. And there will be dietitians who prescribe the specific diets for the bioequivalent study volunteers. And there will be pathologists and lab technicians who carry out all the path lab and clinical laboratory examinations. Then the expertise and professionals in bioequivalence studies continue to be from the bioanalytical background, where the qualifications of the analyst and chemist will be from the pharmacy, chemistry, and life science. And there will be quality assurance professionals. They are also from pharmacy and chemistry, life science backgrounds. Also, the quality control professionals from the same background. And there will be pharmacokinetics officers who are generally from pharmacy background, and there will be statisticians who have the statistics degree or by experience they are qualified to carry out the statistical analysis. Apart from these, we have support functions like logistics and stores maintained by any kind of graduates with relevant experience who maintain all the supply chain management for the bioequivalent centers. Then there will be a support function called HR and administrations, and there the finance and accounts also involved to manage the financial transactions, day-to-day activities carried out in the bioequivalent centers. There will be medical writing department generally being administered by pharmacy graduates or medical professionals, and there will be a business development professionals to bring the business for the contract research organization. So this slide briefly explains the support functions in bioequivalent centers. First, the volunteer recruitments. Volunteers are the main resource for the BE studies, and the recruitment is supported by volunteer recruitment officers, advertisements, and consultants. Then there will be a purchase department which purchases the consumables, materials, chemicals, etc. for the bioequivalent studies. Then the stores, the purchased items will be maintained by the stores, and the inward and outward entries will be carried out, stocks will be maintained for the smooth operation of the BE studies. Then there will be engineering staff who carry out the maintenance of the electrical stuffs, instruments, and equipments in the clinical as well as bioanalytical area and other common areas. And then there will be catering services will be maintained in the bioequivalent center, because the diet is important to be maintained in the bioequivalent studies. Then there will be a housekeeping facility which have the housekeeping staff which maintains the cleanliness of the bioequivalent centers. The support functions continue to be in bioequivalent studies also includes the human resource who maintain the staff welfare, timekeeping, performance appraisals, statutory requirements, and contract staff management. Then there will be a contract staff. So it is not always possible that in-house staff is sufficient for managing some bigger clinical phase of the studies, where we'll have 150 subjects or 200 subjects at a time housed in the facility. So that time the contract nurses, physicians, phlebotomists, housekeeping, etc. will be used for the studies. Then there will be a laundry facility which is necessary for the maintaining of volunteer uniforms, beddings, staff aprons, visitor coats, etc. Then there will be ambulance service which is required for the emergency and critical situations. Then there will be a nearby hospital support for the similar purposes like emergency and critical situations. Then finally the ethics committee which approves the study protocol for the conduct of the study. So to become a operational bioequivalence facility, there will be a lot of regulatory requirements which needs to be furnished before the operation starts. So first requirement is the CDSCO, that is the Indian Regulatory Agency approval for the facility. Then the CDSCO also approves each and every study protocol submitted by the CRO or the bioequivalent center, except the old drugs defined as per the Drugs and Cosmetics Act. Then each and every study will be approved by the ethics committee and monitored by the ethics committee. The protocol will be submitted to ethics committee, which will be reviewed and approved by the committee. And then the import license requirement, where sometimes we need to import the drugs from the foreign countries. So these license will be issued by the CDSCO from Delhi, and the CRO will be applying for the import license from their facility. Then there will be a pollution control board permission for operations of the bioequivalence centers. Then there will be a bio-waste and chemical waste disposal permit or agreement. There will be vendors who carry out these activities. So this should be as per the law and regulations will be carried out by the vendor company. And apart from this, state-wise or country-wise, there will be specific statutory permissions. Anything is required need to be taken for the BE facility before starting the operations. This particular slide summarizes the bioequivalence study flow of activities. So it starts from study design, then the protocol development based on the study design, then the protocol approval by the ethics committee, then the protocol approval by the CDSCO, then if required, we will go for the import license procurement. Then once the license is in place, we will receive the investigational products from outside the country. In case if it is within the country, then we will receive the investigational products without the license. Then there will be a screening of healthy volunteers. Most of the studies are conducted in healthy volunteer subjects. Then the subjects comes into the facility, we do the informed consent process. So once these activities are carried out and volunteers are fit for the study, there will be a check-in process for the study, and then there will be housing of volunteers in the clinical facility. The flow of activities continues like after housing at specified period of time, we will do the administration of study drugs, then the blood sampling, then the sample processing and the storage of the blood samples collected, then the checkout of the subjects. Then once the clinical phase of the study is completed, the bioanalytical method has been developed and validated before the study sample analysis. Then the study sample analysis will be carried out. Then once the sample analysis is completed, the bioanalytical data, the drug concentration data will be generated. That data will be supplied to the pharmacokinetic and statistical analysis. Once the data is analyzed, then the report will be prepared, and parallelly the quality control and quality assurance activities will be carried out. During the entire course of the bioequivalence activities or the operations of the bioequivalence studies, we need to maintain the following practices and principles for the quality of the bioequivalence data what we generate. That includes good clinical practices, good laboratory practices, good documentation practices, and we need to abide by the local guidelines prescribed by various regulatory agencies. So these are the important things which has to be maintained during the operations of bioequivalence studies. Apart from this, we need to follow the guidances subscribed by the specific regulatory agency for which we are going to submit the bioequivalent study, or we are going to submit the dossier for the marketing authorization. So in this particular model, we have seen the bioequivalent study functions, how it works, what is bioequivalent study, who are the professionals involved in the bioequivalent study, their qualifications, and the general flow of activities, etc. In the upcoming modules, we are going to see in detail how each and every function is going to be operated in the bioequivalent centers and what are the scientific principles involved in the bioequivalent studies. Thank you.